Professor DaniZamir is Emeritus Professor for Plant Breeding at the Hebrew University of Jerusalem in Israel, where he started working almost 40 years ago in 1982. In his research, he combines classical and genomics-assisted methods. He has established a unique germplasm collection on tomato, which has also been used in the TOMRES project. Dani has won many national and international awards for his research, and in TOMRES he is the co-leader of Work Package 1 that Andrea Schubert has just introduced.
So I will take another angle on the complexity of drought, and that's the genetic angle. We'll tell you about using exotic genetic libraries for drought tolerance in processing tomatoes. Next slide, please. So I'm a student of Dr. Rick, who did a lot of collections early on, prior to 1992, I think. And he collected a lot of wild accessions from the genus Lycopersicum, or tomato now, Solanum lycopersicum. And he loved South America, and you can see him with the children there. He would repeat and come back to the same places year after year. And a lot of the collections that he made, if you go back to the place where they collected, you'll find parking lot, everything, no, or not everything, but all these that are in agricultural area and cities were lost. Fortunately, he left us many wild species, accessions, and it's always like this for me, but in science, when you don't know what to do, so you make another course. And the next slide will show you one of the courses that we made. It's from Solanum penelli. It's a desert plant, next slide, that can easily cross to the cultivated tomato. So you see the cultivated tomato, the F1 hybrid that you see in the middle is much stronger, much more vigorous than its two parent, and it's in fact heterosis that we see there. Our lab was interested in the genetic basis of this heterosis. Next slide. So in order to map it, we developed introgression lines where on top we have the chromosomes cartoons of the 12 pairs of homologs of the wild species. Underneath it, in red, the M82, which is the cultivated tomato varieties, and then a set of 76 lines, each carrying a single segment from the wild species. So it's easier to do the mapping. If you find a difference between M82 and IL11, you can say that the gene that is responsible for this difference resides on the northern part of chromosome 1 of Solanum penelli. This was a very powerful tool, but it had the disadvantage. We know also from the previous talk, Francesca, that life is complicated and genes do not work alone. Next slide. We know that there is a lot of interaction between the genes. So on the left, you see the pegs are the genes, and the topography, the mountain, is the topography. So on the left, you see one gene pulling along and shaping the topography. But when you look at all of them, next one, you can see that in many cases, 3 or many more genes interact with each other to pull on a phenotype. The introgression lines do not allow one to identify such interacting factors because it has only a single segment coming from the wild species. So in order to be able to look at epistasis, we developed a new population, next slide, that that allows us to zoom in on epistasis. The reason that it's difficult to analyze genetically, to analyze epistasis, is that most segregating populations that we study is about 102 or 200 individuals, and one needs thousands of individuals to have the capability to map interacting QTL, interacting genes. The other thing is that when you analyze statistically the data, basically what you're doing is you analyze the phenotype, or you analyze each marker with all the rest of the markers in the genome. And then the second markers with all the rest of the markers of the genome. So you do millions of comparisons, and then you have to compensate for the multiple testing penalty, and then nothing is significant. So that's the difficulties to uncover epistasis. Has a small population size and also the multiple testing penalty. Next slide will show the population that we developed prior to the TOMRES project, where we took Solanum penellae, call it the lost accession. We crossed it to determinate tomatoes and indeterminate tomatoes, and we crossed the two tomatoes together. And I will focus today about the cross of Solanum penellae to a determinate background called Lea, where we had 1,500 backcross 2 self-6 individuals that were all genotyped thanks to the team of Giovanni Giuliano in the G2P Soul project. Okay, the population was again genotyped, and we can see the maps of all the chromosomes. On the y-axis is the distance in base pairs, and on the x-axis, sorry, on the x-axis is the distance in centimorgan. The y-axis is the physical distance. You can see that there are two regions for each chromosome. Let's take chromosome 1, for example. On the left, there is a lot of recombination for each physical distance. And then in the centromere, there is little recombination all the way to that's where you are. And then on the long arm of chromosome 1, again, there is a lot of recombination. And we have that for the rest of the total genome. We have cases like on chromosome 7, where we have these jumps in steps in comparisons of the genetic and physical map. Seems like regions of hyper high recombination or something else that we don't understand. Anyway, we don't know what it is. Next slide. So we planted these populations both as inbreds, backcross 2 self-6, so plants that are homozygous for progressions coming from the wild species, and the hybrids of this plant with the cultivated tomatoes. So we have on the left the Lea bills. We have homozygous lines of homozygous for the introgression, and on the right, the Bill hybrids is the same lines heterozygous for the introgressions, and we had a number of replications, and we planted them out in a field that had no irrigation. So we gave about 10% of the amount of water during transplanting of the seedlings, and that's it for the rest of the season. And here the single plants that we harvest and get yield data for. Next slide. And you can see that we did it in different backgrounds, 4144, and another experiment 4144, and then another experiment in the hybrid. And when you do this is the size of the effect. The size of the effect relative to the mean of the population. So you see that there is a region in the genome that highly significantly increases yield in all courses. And the increase is about 15%. There is also another one that is consistent on the other ones that are consistent on different chromosomes. So we can map QTL that increase individually, not epistasis, increase yield. The next slide shows the epistasis analysis, where we do the analysis, we can find regions where, next move, you can see a region in comparing the two maps that is bright. This is a region where there is an epistasis between chromosome 7 and chromosome 1. And then if we go to next slide, we look at the values of the genotype. So you can see that on the left, 0, 0, these are the individuals that do not carry chromosome 1 or chromosome 7. And this is the mean is 3.5 kilogram per plant. On the right is IL12. On the right of it, there is 1, 2. So the plant does not carry chromosome 1, but it carries chromosome 2. And then chromosome 2, 1, the next one, all of them have the same, about the same value of 3.5, 3.6 kilogram per plant. But the ones that are 2, 2, that are double heterozygotes, you see that the yield is significantly higher. So here we have a case of epistasis. Chromosome 1 alone doesn't do anything. Chromosome 7 alone doesn't do anything. But when you put them all together, they do a gig. So that's nice. But of course, one has to validate it. And next move, you could see the validation that was done last year. This time, under 3 irrigation regimes, which is, we call it 0, but in fact it's about 10%. Then in the middle, 50%, and on the right, 100%. And you can see that in all these trials, the individuals that are heterozygous for both introgressions have higher yield than the rest. So it leads to the effect of these two introgressions on productivity. Next slide. So one has to be lucky every once in a while. So as we developed this population and worked hard to make all the backcrosses and the selves and keep track of everything, and there came this new disease of tomato that is called tomato brown rugose fruit virus that is very strong in Israel, in Mexico, some cases in Europe. You see the symptoms. well, turns out that the wild species that the Penelli that we worked with was resistant to the virus, and we could map the gene for resistance to the map. It falls very close, or possibly is the TM1 gene of the tomato, the classic TM1 on chromosome 2 of tomato. Next slide. So I showed you today what I did in TOMRES project. All the phenotyping was done according to specifications and consultations with the members of the project. And now I am continuing this research because the work is not yet done in a HarnessTom project that Tony Granell is running. So in this project, I think there was a year and a half after the start of the project, we were hoping to map the gene. So we mapped the gene already, so we could do more delicate genetic analysis, and it will be fun. And the contribution of the TOMRES project is essential for this research. And I thank Turgeman, who's the PhD student that did all the work. And I thank you very much for listening.
Thank you very, very much, Dani, for this really interesting presentation. And especially also for your last slide showing how TOMRES research feeds into other and future projects as well. I think that is wonderful.